All right. We are back again. So lesson 9 is on neural pathways. And we can really think of this as neural pathways, neural systems, neurotransmitter pathways. There's a few different ways we can talk about this, but it's essentially the same thing. What we're going to be talking about is certain pathways in the brain that are regulated by certain systems of neurotransmitters. So for example, you might have one region of the brain that consists of a whole bunch of cell bodies of a specific type of neuron. Let's say it's a dopaminergic neuron, so that means that it makes dopamine. Well, what happens is these neurons actually will project to different regions of the brain, right? And send projections to the spinal cord as well, okay? But really we're going to be talking about the ones throughout the brain here. And so depending on which neurotransmitter system, which neural pathway we're talking about, we're actually talking about different types of behavior that's going to be regulated. So that's what we're going to be exploring today. And I can't really think of a better way to start this out with our discussion of neural pathways than to visit one of my favorite television characters, Sheldon. You may know, be familiar with the Big Bang Theory. So what I would like you to do is watch the following clip here, and then we're going to talk a little bit about what's happening here. And as you're watching this clip, I want you to ask yourself a couple of important questions. First of all, what is Sheldon doing in this particular scene? What is he trying to do? And does it work? Is this a technique that we can actually use? And how is he shaping behavior? And then the second question, is there a brain pathway that mediates these particular types of responses?
Are you finished?
Well, thank you. How thoughtful. Would you like a chocolate?
Yeah, sure. Thanks. Oh, sorry, Sheldon. I almost sat in your spot.
Did you? I didn't notice. Have a chocolate.
Thank you.
You're here a lot now.
Oh, am I talking too much? I'm sorry. Zip.
Thank you. Chocolate?
Yes, please. Oh, hey, Kim. Yeah, you know what? Hold on. Let me take this in the hall. You'll never guess who they got to replace you at work.
Okay. I know what you're doing.
Really?
Yes! You're using chocolates as positive reinforcement for what you consider correct behavior.
Very good. Chocolate?
No, Sheldon, you can't train my girlfriend like a lab rat.
Actually, it turns out I can.
Well, you shouldn't.
There's just no pleasing you, is there, Leonard? You weren't happy with my previous approach to dealing with her, so I decided to employ operant conditioning techniques, building on the works of Thorndike and B.F. Skinner. Yet by this time next week, I believe I can have her jumping out of a pool, balancing a beach ball on her nose.
No, this has to stop now.
I'm not suggesting we really make her jump out of a pool. I thought the bazinga was implied. I'm just tweaking her personality, sanding off the rough edges, if you will.
No, you're not sanding Penny.
Are you saying that I am forbidden from applying a harmless, scientifically valid protocol that will make our lives better?
Yes, you're forbidden.
Bad, Leonard. You know, using positive reinforcement techniques, I could train that behavior out of her in a week.
No.
If you let me use negative reinforcement, I can get it done before we go to bed.
You're not squirting her in the face with water.
No, of course not. We're talking very mild electric shocks. No tissue damage whatsoever.
Forget it!
Oh, come on! You can't tell me that you're not intrigued about the possibility of building a better girlfriend.
I'm not! Penny's qualities, both good and bad, are what make her who she is.
You mean like that high-pitched, irritating laugh?
Yes.
You wouldn't prefer a throaty chuckle?
You're not changing how Penny laughs.
No, that would be incongruous. I was going to lower the whole voice to a more pleasing register.
Sorry, guys. That girl is freaky!
Come again?
Freaky.
Freaky?
Yeah, freaky.
Have a chocolate.
Thank you.
Well,
Pretty awesome, huh? So, if you guessed reinforcement and reward, you are 100% correct. So really what Sheldon was doing, in that scene in a very funny way, of course it's great, it's one of my favorite scenes, is he was trying to shape behavior. So he was trying to shape behavior by activating a very specific neural pathway, okay, or a neurotransmitter system in the brain that is responsible for reinforcing actions and behaviors. So that is to say, we've all seen the sort of situation where maybe your dog jumps up on the couch, and maybe you have really nice couches, and maybe you don't want your dog to be jumping on your couch, so you spray the dog in the face with a bottle of water. Now, I don't particularly subscribe to this method of reinforcement, but I've seen it done before, okay? So that would be introducing a stimulus that's going to produce a negative effect and is going to decrease the likelihood that that dog is going to engage in that type of behavior in the future, right? Similarly, you may get rewarded for getting A's in a particular class. Let's say, for example, your honors neuroscience class. And maybe you have a family member who pays large sums of money for each A that you get on your report card. Now, that would be a mechanism of positive reinforcement where you're actually given a stimulus right after you perform some sort of act, and that is supposed to increase the probability that you're going to engage in that behavior in the future, i.e., study very hard, do your homework, etc., so that you can get the A in the class. But really, as a neuroscientist, we're less interested in the monetary benefit than we are in the actual pathway that underlies this sort of phenomena. And in the brain, this particular type of reinforcement pathway is regulated by a dopamine system. So here you can see on the left side, we have a presynaptic neuron. So we talked a little bit about this last time. And we have a postsynaptic neuron. And on the postsynaptic neuron, we have dopamine receptors. And in the presynaptic neuron, we have vesicles filled with dopamine. So if we take a zoomed in view at the synapse, we know that in response to a certain stimulus, this neuron is going to fire, okay? And it's going to release dopamine into the extracellular space. That dopamine is going to bind to the dopamine receptor on the postsynaptic cell. It's going to cause a response in the cell. Now, once the dopamine's done working, it's either going to get taken up in the cell, okay, into one of these reuptake transporters. By the way, what's this one called? Cricket. DAT, right? DAT, dopamine transporter. That was just a little quiz for you. So, there are a couple of different dopamine pathways in the brain. We're going to talk about each of them, but the one that we just visited, the one that Sheldon was trying to tap into, is actually called the mesolimbic dopamine pathway. And if we look at this brain on the left, what we can see is that there's a few different colors here and a few different what look like branches, right? So this is sort of like a tree that's branching out. And we can see that the blue is serotonin, just happens to be in this picture. I like the picture, so I used it. But forget about serotonin now, because we're going to talk about that separately in a minute. All right. But the red here, you see we have dopamine pathway, and this says nigrostriatal. We're going to talk about what that means in a minute. This green one here says mesolimbic. Now, this green dopamine pathway, so these are actually represent neurons. The cell bodies originate over here, and different dopamine neurons actually project to all these different regions of the brain. Okay, so this is what we mean when we talk about a neural pathway. So they're sending projections, their axons are spreading out, and they're synapsing in different parts of the brain. And yes, they're going to synapse with different types of neurons. And yes, they may not be dopaminergic neurons, they might actually be something different. Okay? And here on the right side, this is just a very simplified version, okay? We have a couple different brain regions here that we're going to talk about, and these are essentially important brain regions in what's the best way to say it? Reinforcing any kind of behavior, right? So anytime that you feel pleasure or anytime you notice something that's really important or in your environment, things of this nature, you're going to have this particular pathway activated. This is the mesolimbic pathway. Interestingly, same pathway that's activated in response to drug use. And we'll talk a little bit about that in a little bit too, okay? So, but what's really neat about this is that for us, you can take this at face value because you could say, okay, Dr. L is telling me all about the brain, and he's a neuroscientist, so he probably knows what he's talking about. But how do I know? Right? And how does anybody else know? Well, the reason that we know is because we have done some very interesting and clever experiments using animal models. Now, we talked a little bit about animal modeling. Of course, if we're going to use an animal model, there's several different ways of going about this, but if you think about it, we could ask the mouse itself. So let's say, for example, we're going to give the mouse a treat after it runs through a maze. Okay, we can ask the mouse, did you like that treat? You think you're going to be able to find the end quicker next time, right? But we can't, the animals don't really talk back, okay? So it's not the best strategy, really, to try to find underpinning, neurophysiological behaviors, or the substrates for which, or we can actually observe the animals. And what scientists have done is some very clever experiments whereby they have produced certain types of stimuli that are either rewarding or aversive, and they have watched the behavior of rodents. And they've actually tried to train them to do certain types of tests that they otherwise would not be interested in doing, okay, by giving them some sort of pleasurable experience after they do the task. Now, I could talk about this all day, but I think this is something that's really better observed. So what I would like you guys to do is actually watch this video clip, and then we're going to get back to the reward systems. Now, what you're going to see in this clip is you're actually going to see an experiment where animals had electrodes implanted into their brains. And it wasn't just randomly implanted, okay? They were implanted into a very specific part of the animal's brain. And they received a very mild electrical stimulation. And we remember that neurons work both chemically and electrically, right? So if we provide a weak electrical stimulation, it will actually cause the neuron to fire, and it will have an action potential. So what they're essentially doing are stimulating electrically, right, different parts of the brain to see whether or not the animal is more or less likely to engage in a certain type of behavior. And I'm going to let you guys figure out the rest.
You almost look like you're smiling a bit. Are you?
Oh, no, I might.
He's implanted electrodes in the brains of chronically depressed patients, hoping to stimulate pleasurable sensations.
I guess I've cracked up all the way. I don't know.
I mean, that's the reason you're smiling.
Probably.
Or you look like you're really... What are you laughing
I
about?
don't know.
Huh?
Are you going to do something?
Not surprisingly, the experiments were controversial, but the electrodes did have an effect, especially near an area of the brain called the nucleus accumbens. Heath even let his patients control their own stimulation.
I find this button the best.
That's number 2 button.
Most pleasurable.
Yeah.
Perhaps we get pleasure from drugs because they stimulate the same brain areas as sex and food. Working in Montreal in the 1950s, James Olds devised a crucial test. He worked out a new way to, in effect, ask animals how they felt.
This is a device which trains the animal to turn on the stimulus for himself. Here you notice the animal wandering toward the pedal. When he first touches it, he gets no stimulation. It's not connected yet. Then, one time he touches the pedal, it does turn on the stimulation. You can see the lights go on.
Now, the interesting thing about the old stimulation, the rewarding stimulation, was the strength of it. It was somewhat different than rewarding stimulation for food and water in that there was no satiation. Animals would continue for hours and hours and didn't seem to satiate. They didn't feel full. They would just keep doing it for long, long periods of time. They would do it to exhaustion.
Now, I will electrify the grid. The animal must cross an area that gives a very painful shock to the feet in order to get to the pedal and stimulate the brain.
And a rat wanting brain stimulation would brave a shock stronger than even a starving rat would brave to get food.
This shows a rat is willing to pay a very high price in order to get to the pedal.
Human addicts will pay just as high a price for their rewards.
The fascinating thing was that we found in our laboratory that every single drug that increased sensitivity of the animal to brain stimulation was either an abuse substance or a substance that has potential for abuse.
So James Old's technique of the 50s gave scientists a way of testing for the addictive potential of drugs, and a way to find out where in the brain they acted.
And I think there's general agreement that this is the area where the action is in terms of the rewarding effects of drugs, whether they be heroin, whether they be amphetamine, whether they be cocaine or angel dust or whatever, any drug that produces rewarding effects, it's believed this is where the action is.
Using similar methods, Chris Fibiger and his colleagues in Vancouver wanted to know which pleasure areas are active when we crave both drugs and food. They found that the area Heath stimulated in his patients, the nucleus accumbens, is the center of a key network, the dopamine circuit. Heroin seems to act in one way. Cocaine in another. And both hit the nucleus accumbens, which the Canadian scientists recently proved is also active when we crave food.
If we selectively take these dopamine neurons out of that part of the brain, the animal loses interest in self-administering cocaine, it seems. Apparently, the drug is no longer reinforcing. The same story seems to apply for amphetamine. If you destroy these dopamine-containing neurons in the nucleus accumbens, amphetamine self-administration disappears as well.
A heroin addict has volunteered to be given a large dose of morphine, chemically similar to heroin, by scientists at the National Institute for Drug Abuse in Baltimore.
Relax.
Today, using new methods, researchers can at last start to look at where drugs act in the human brain. The new machines reveal why different drugs make us act like primitive animals. Morphine shuts down the higher cortex and leaves the older emotional brain in charge. Cocaine gives a boost to the whole brain, but especially stimulates the primitive centers of emotion. So from those rough and ready experiments in the 1950s, scientists did learn where in the brain many addictive drugs act.
All right, so that was pretty awesome, right? Really interesting experiments. Now, these Olds and Milner, who I don't think they mentioned Milner, but he was also instrumental in those experiments. You know, kind of not so great if you're Milner, getting your name left out like this. And it does happen in science, but very important experiments in the 1950s. It actually helped us to understand the structure-function relationships such as these in the brain, which are so critically important. Now, dopamine pathways, okay, we already sort of saw this, are super important for a few different reasons. And it's not really just about reward and reinforcement, which is what we talked about a little bit so far, right? So these are what we know of as the mesolimbic, okay, or the mesocortical dopamine pathways. Now, sometimes people actually will say mesocorticolimbic because they will sort of incorporate all of these pathways and kind of mesh them together. Because if you remember from our earlier discussion about the brain, the cortex, and particularly the frontal cortex, and sometimes it's called the prefrontal cortex, is even more of a specific region of cortex. It really receives inputs from everywhere. And a lot of it is actually what we call association cortex, which means that it receives and sends signals to different various parts of the brains for all kinds of different things, okay? So the cortex is sort of involved in everything. It's kind of all over the place, and it's got its hands everywhere, all right? But there are two key projections here, the mesolimbic and the mesocortical, and what they do is they basically go from the midbrain region, okay? Now this VTA stands for ventral tegmental area. So this is one of the key areas in the brain where dopaminergic neurons actually arise. Okay, so this is a site where dopamine is produced. Remember, it's produced in the cell body of the neurons. All right, and this structure here is called the nucleus accumbens. And as you know from watching the video, the nucleus accumbens really has sort of over the years become known as the pleasure center of the brain. Now, it's not that simple, of course, right? Because it's not really just about pleasure. It's really about reinforcement. So even if you were to get, as opposed to, say, receiving chocolates, all right, even if you were to get whacked over the head with a mallet, you would still have a large release of dopamine in the nucleus accumbens, okay? So this is actually an area that's very important for signaling to you that something is important, right? And when things are important to us, a lot of times we want to do them again, so our brain has a way of rewarding us for doing them, i.e. we feel good, okay? But it also, sometimes we feel bad if it's something that we don't want to do again. So that's sort of a double-edged sword, right? Now these pathways actually are involved in the addictive properties of drugs as well. Because any drug of abuse is essentially going to activate these same areas in the brain, and you saw that from the video too. So these areas in the brain respond to drug use because dopamine is released. And it's not just dopamine, okay? There's actually a whole bunch of different neurotransmitters that are released, but this is the one area that's sort of known for that and converges as such. ADHD is also another sort of area where we have found that dopamine signaling is important. So this is something that's a little bit newer, okay? We haven't known about this for as long as we've known about addiction and about the reinforcing properties of different types of stimuli, okay? But aside from that, we also have this one called the nigrostriatal region. Now this nigrostriatal pathway, okay, is called this because it actually starts out with dopaminergic neurons that originate in an area called the substantia nigra. And we're going to see that it's here, okay? So here you have your VTA, that's the ventral tegmental area, that's the mesolimbic pathway. Then you have your substantia nigra, very, very close by, okay? But it's actually, this is what is more of a motor loop, okay? So dopamine's really important in motor function. And if you know anything about PD, or Parkinson's disease, one thing that you'll note is that one of the things that became evident as we learned more and more about Parkinson's disease is that a hallmark of Parkinson's disease is the loss of dopaminergic neurons. So for some reason in the brain, neurons that make dopamine, okay, specifically, dopaminergic neurons begin to die off, okay? And they begin to die off in the substantia nigra. And because this is a motor pathway that's very important for motor function, you see the motor dysfunction and deficits in Parkinson's patients, right? And in fact, one of the things that was done early on, if you ever get the chance, you should try to watch the movie Awakenings. Very good movie about Parkinson's disease. individual has Parkinson's disease. And one of the early treatments that they tried to use was actually to give people the precursor for dopamine called L-dopa or levodopa. Now, if you remember from the last video, we know that L-dopa is synthesized in that biosynthetic pathway right before it becomes dopamine, right? Now, the problem is that dopamine doesn't really cross the blood-brain barrier very well, but L-dopa does. So in this way, they could actually give the patients L-dopa and try to boost the amount of dopamine that's available. And it worked for a little while, but not in the end. It turned out to be still a lot of work needs to be done to find a way to cure Parkinson's disease. The last one is the tuberoinfundibular pathway. This one we're really not going to talk about too much because it's a little bit more specific. You can see it's a kind of a short projection here. Down here at the base of the brain and the ventral surface, you have this pituitary, okay, the pituitary gland. Now this is an endocrine gland that's, super important. It actually regulates lots of stuff. But one thing that it does is it actually, and one thing that the dopamine is important for is regulating the release of prolactin. And prolactin is sometimes thought of as a pregnancy hormone because it's one of the hormones that gets released to help, the female produce milk when there's a pregnancy and needs to be able to feed the young and things like that. Okay. So here, nucleus accumbens and ventral tegmental area. These are the two dopaminergic regions, okay, sources that are really important for reward and reinforcement. So that's something that you're going to want to remember. And the substantia nigra, okay, is the heart of the nigrostriatal pathway. And this is associated with Parkinson's. And we can think of that because we can think of motor behavior, okay? So we're going to go through the other neurotransmitters and kind of just point out some of the important things about them. We're not going to spend tons of time talking about, each little projection because as you can see from this diagram alone, it's tremendously complicated, okay? Neural pathways are things that people study for years. I mean years, decades. An entire body of life's research into understanding how neural pathways function. All right, so we're kind of just going to scratch the surface and point out some of the interesting things. So serotonin, the raphe nuclei. So if you look at the brainstem down here, there's actually a few different places. Now, you probably think of nuclei as being a bunch of different cells, right? But in the brain, anywhere where you have a whole bunch of cell bodies that are kind of in a big cluster together, that region of that cluster of cell bodies is called a nuclei. Okay, so what this means is that there's these different clusters of cell bodies of neurons, all of which are producing serotonin. Okay, so these serotonergic neurons originate in the raphe nuclei. All right, now what does serotonin do? Serotonin is perhaps most well known for its role in depression, also in regulating sleep patterns and things like that. So not surprisingly, the serotonergic pathway has a lot of projections to structures that we call limbic structures, okay? Now, the limbic system of the brain, we're going to see that in a minute, is really an area of the brain that's associated to a large degree with emotion, okay, affective states. So affect, A-F-F-E-C-T, not effect, okay, but affect meaning emotional state, mood, right? And also learning and memory. So there's a lot of structures in there that are associated with these things. So serotonin goes to a lot of those places. It's also involved in signaling for nociception, or pain as it's commonly referred. So you have these serotonergic innervations. So these serotonergic neurons, some of these nuclei are going to project to the spinal cord. Spinal cord is super important for pain signaling, right? And if we take a closer look at the limbic system, the more we look into it, the more we realize that not everybody really agrees on exactly which structures are in the limbic system, but what we do agree on is that it's sort of an quote-unquote old brain. And I say that because I always think expressions like this are sort of silly, but this one makes a little bit more sense. Because if you look at the brain, you can see we have all this. Remember, we talked about the development of the brain earlier on and how the cortex, right, the prefrontal cortex really is signifies the evolution of the human brain and how it really evolved and grew. But these more primitive, like midbrain and hindbrain areas here, are what are thought to have been the old brain, okay? So not only was this maybe here first, because they're super important structures, but they're very primitive structures, meaning that they're involved in processes, right, and functions that are necessary, very, very necessary and vital to life itself, right, for the species to continue to procreate and to survive, really. Okay, so you have these different regions. This is the amygdala. This is really the seat of emotion. Okay, so anytime that you think about somebody who is having problems with maybe anger management, or you might have heard some stories about people who had damage to a certain part of their brain over by their amygdala, or neurons that project to the amygdala, and they actually have trouble regulating their behavior. Okay, they get very overactive and over emotional. So we think of the amygdala as being a center for that. The hippocampus, as we mentioned before, is really a memory center. So if you think about the way that the very fundamentals of being able to survive for a long time, being able to, find a food source and being able to, you know, do all the things that a species needs to do to survive, it would make sense that we would have a memory center involved. We would have some sort of really sort of an emotional center here, and of course the thalamus and the hypothalamus. Now remember, the thalamus is kind of a way station. It receives inputs from all over the place, so it's kind of like a central hub in the brain, and the hypothalamus is involved in controlling a lot of vital functions. Okay, so these structures together, it makes sense that this would be kind of an old brain, because they're things that are really super important for any organism. We would have needed these. Okay, now this cortex is kind of the newer part, because this allows us to engage in a lot of advanced reasoning, logic, planning, things like that. Things that maybe aren't so completely necessary, right? It is important to be able to survive. It's less important to be able to ponder the origin of our existence, right? So that's kind of the way that you can think about it there. So different kinds of structures in the limbic system. Well, I sort of talked about these already. There's one I didn't mention, which is the basal ganglia. Now, your basal ganglia is actually a region that's seated way deep inside here, and it includes an area called the striatum, which is actually really important for movement. And we'll talk about this a little bit more as we go on, so I don't want to get too much into that yet. But just know that this limbic system is important. So all of the neurotransmitter pathways that we're going to be looking at, they send projections in some way or another to regions of the limbic system. Okay. The dopaminergic system that we looked at before actually does send projections to the limbic system. Now, the nucleus accumbens is right in that striatal region, the striatum. So that's really a basal ganglia structure, okay? And we didn't talk about that before because we didn't really know what it was yet, okay? But the nucleus accumbens, okay, and that motor loop of the nigrostriatal dopamine system, those neurons project actually to basal ganglia structures, okay? So take-home message, limbic system is super important. It's important in shaping behavior. It's important in reinforcement. And what better way to reinforce something than to make sure that you remember it, right? And to make sure that it has some kind of emotional component. So these things all kind of fit together. The next pathway is the cholinergic system. So cholinergic means acetylcholine. So we're talking about the neurotransmitter acetylcholine here, all right? And these neurons actually are going to project out of the midbrain region. So remember, the tectum and the tegmentum are the two regions of the midbrain, okay? Here we have the basal nucleus of Maynard, okay? This is actually what's referred to as the basal forebrain. So you remember the forebrain is sort of goes up to where you have the tectum and the tegmentum would be in here. That would be the beginning of your midbrain, and your hindbrain would be back here, right? Okay, so this is the basal forebrain, basal because it's near the bottom, okay, and forebrain because it's actually in the forebrain area. So this is where we have the origination of cholinergic neurons. they go lots of different places, okay? So the cortex, the hippocampus, for example. One of the cool things about cholinergic pathways is that aside from the ones in the brain, okay, acetylcholine is super important at neuromuscular junctions. And I think we talked about this a little bit more, a little bit before when we first introduced the neurotransmitter. Okay, so these neuromuscular junctions where you have acetylcholine release, this is actually how a lot of these paralyzing venoms and toxins work. Okay, because either what, and we're going to do this specifically when we do the pharmacology section, we're going to look at some examples of this. Okay, but this is kind of just to introduce it a little bit now. If we have a lot of acetylcholine in the synapse and we're giving a toxin or a venom that actually blocks it from being reuptaken, sometimes what it can do is it can keep activating the postsynaptic cells so much that it shuts off. Okay, and this shut off causes a problem. Right. Similarly, restricting the amount of acetylcholine that gets released can cause paralysis. So there's different ways that we can actually affect this system to kind of cause problems. Right. Cool thing also about acetylcholine, right, and cholinergic pathways is that there's a lot of interneurons that are cholinergic. Okay, now what do we mean when we say an interneuron? Well, it's exactly what it sounds like. If you were going to have to travel a long distance, you might want somebody in the middle, okay, to kind of take some of the load off. So sometimes you can think of it as a baton race. So you have two people running all the way around the track and they have to hand the baton off to the other person. Well, if you've got somebody in between, it's a little bit less that you have to run, right? I think we saw some of these interneurons when we were talking about the sensory inputs and outputs, so the efferent and afferent connections, and you sometimes have a neuron that synapses in the middle of the two. Okay, these are interneurons. Well, we also have interneurons in the brain. Okay, and in that striatum region, remember that's the basal ganglia, the striatum, which is involved a lot in motor function. We have interneurons there. We also have a lot of interneurons in the cortex. So one way that we can sort of fine-tune the way that signals are sent and received is by changing the amount of cholinergic signaling, okay, the amount of acetylcholine that's being released. So in addition to its role at the neuromuscular junction, it's important for learning and memory. It's important for arousal. So anytime that you have to focus and concentrate, you're trying to remember something, acetylcholine is there. Interestingly, Alzheimer's disease, I think most of you know that Alzheimer's disease is a disorder that involves loss of memory, right? And so one of the things that researchers have found over, the last, I would say probably a couple decades of doing research on Alzheimer's disease, both in humans and in animal models, is that one of the problems with Alzheimer's disease seems to be that there is a loss of cholinergic signaling. Now, this has been reported a couple of different ways. It's been reported as a loss of cholinergic neurons, right? So just to remind, that means neurons that produce acetylcholine, or loss of cholinergic receptors, all right? So there's two ways that we could decrease the amount of cholinergic signaling. We can either reduce the number of neurons that are making acetylcholine. So in this way, it's kind of similar to what happens in Parkinson's disease, right? Where we reduce the number of dopaminergic neurons and it causes problems with motor function. Well, here, reducing the amount of acetylcholine producing neurons can cause some problems related to memory. The other way we can do this is even if we have the same amount of acetylcholine being produced, if we reduce the number of receptors that it binds to, right, then we're going to decrease the magnitude of the effect when acetylcholine is released into the synapse. And so it's sort of two different sides of really the same coin. What we're doing is we're reducing cholinergic signaling. So this is something that's been documented and something that I'm sure will continue to be visited over time. The last pathway is the norepinephrine and epinephrine pathway, and we're going to focus more on norepi for reasons that will become evident shortly here. So important points about this pathway, again, before we go into more detail later, right, just to kind of get the basics down. We have this structure here called the locus coeruleus, okay? No, Gladiator fans, it is not Marcus Aurelius, Okay, this is the locus coeruleus, and this is a center for the production of norepinephrine neurons. All right, they go lots of different places, notably to, again, limbic structures like the thalamus and the hypothalamus, okay? Also to the cerebellum and to the cortex. Now, almost basically everything projects to the cortex. Like I said, it's kind of involved in everything, so that's not really any kind of big surprise there, okay? But what it actually does do is it projects to the cerebellum, so can play a role in things like motion, things like that. But what's cool about norepi is that norepi is actually super important, okay, in what we call responsiveness. And by responsiveness, I don't mean answering when somebody calls your name, but what I mean is that when you actually are introduced with new stimuli in a familiar environment, okay, your ability to attend to it and recognize it, okay. Now this seems to be a skill, really, for lack of a better word, that is highly dependent upon norepinephrine signaling, right? And this has been shown because basically what they can do is they can hook up different devices to your scalp, and they can measure the amount of blood flow in your brain in different regions when you're being presented with different types of stimuli, okay? And by doing this, they can look to see which regions are activated at different times. And what they've noticed is that when human subjects are presented with something new, okay, a new stimulus, something they haven't seen before, norepinephrine pathways fire more rapidly and for longer periods of time. So that's pretty cool, right? Autonomic nervous system functioning, it's super important. I think we talked about this before. Norepi and epinephrine basically project and send projections and get released all over the place in the body, okay? But autonomic function is important. Here, I didn't include something for epinephrine because epinephrine is more of a peripheral sort of neurotransmitter, okay? In fact, most of the epi is produced in your adrenal medulla gland, which is why we don't really have a schematic here where we're going to show epinephrine signaling in the brain, but it actually is made in a couple of different places in the brain. But for now, we're going to kind of sort of ignore epinephrine for now, adrenaline, and we're going to talk a little bit more about norepi in this class, okay? So there's our lecture on neural pathways, and I hope you liked it and you got to watch a few cool videos, and you can take this quiz, see how you did, and I will see you next time.